Hi, everyone. My name is Matthew Frigault, and I'm an assistant professor and a clinical director of our cellular immunotherapy program at Massachusetts General Hospital. And I am excited and thankful to be here to discuss an abstract from ASCO, which is abstract 8015, which is a phase 1 study of CART-ddBCMA in relapsed refractory multiple myeloma. So I'm really excited about this abstract. CAR-Ts and multiple myeloma have been showing a lot of potential. We just had a recent FDA approval with idacaptogene dicloglucil, which is the very first BCMA-directed CAR-T cell therapy for multiple myeloma. And the interesting thing with this class of drug is that it's actually a living drug. It's derived from a patient's own immune system. So it utilizes the T cells, which naturally fight off infections and even cancer in some instances in the body. And we genetically modify those cells to become hunter killers of anything that expresses a billboard that is expressed on multiple myeloma, in this case, BCMA. So as I mentioned, we already have one FDA approved product, and we have other products like cilta-cel as well as ddBCMA that are currently in development. And the interesting thing about this product that we presented data on, I'll go through the results, but are that it's one of the first times we're using a completely synthetic binder. So usually when we make CAR T cells, these things are derived from antibodies that can come from mice. In some instances, they're actually derived from llamas, or they're called camelids, but they're a foreign protein that's not usually seen in human beings. And as such, that foreign protein can cause the immune system of patients to recognize it and actually to try to reject the CAR T cells. So this new novel binder that again, it is foreign, but it's been engineered specifically not to cause any kind of immune response. And it's also very, very stable compared to some of these other components. So what we've seen is we're seeing very high expression and stable expression of the CAR or the ddBCMA component on patients' T cells. And so this is a phase 1 dose escalation study, meaning we did 2 different dose levels, and it involved collecting a patient's T cells, manufacturing the CAR T product specifically for them, giving them low doses of chemotherapy to what I say open up space within their immune system for the CARs to get on the immune system highway and to grow and to do what they need to do. So when we looked at our study here, we have 16 subjects who are enrolled, and we had 13 subjects treated. 1 patient wasn't yet available based on the timeline. 2 patients were pending treatment, and 1 patient had to discontinue because of an adverse event unrelated to the treatment prior to cell infusion. And across the cohorts, we did 2 dose levels. We did 100 million CAR T cells, and we did 300 million CAR T cells. And what we can see is that we had a median age of 73 and 60 years old. So in a little bit of an older population compared to some of our other studies. And in the majority of cases, most of these patients had high-risk features. So about 9 out of the 10 that we could evaluate had high-risk features by cytogenetics. The majority of patients had high tumor volume inside of their bone marrow, and they had around between 4 and 5 prior lines of therapy. Most patients had a prior bone marrow transplant, and actually around 10 out of the 12 patients that were treated actually were penta-refractory, so the prior lines of therapy. So that included, multiply refractory to IMiDs and PIs and anti-CD38 targeted therapy. So these are a highly refractory group of people. Now, it being a phase 1 study, the first primary outcome is actually to look at safety, to see what kind of adverse events there were and make sure it's safe going into people. And what we can say is that we only really had 1 severe adverse event, which is considered grade 3 ICANS, which is a type of neurotoxicity that can cause confusion, sleepiness, things along those lines, and can be life-threatening. But to put that into context, in some clinical trials, we see grade 3 ICANS or higher in over 35% of patients, especially those with leukemia and lymphoma. So this is very much well in a safe range for what we'd expect. And in this entire cohort of people, we used our typical management strategy for CRS like tocilizumab and steroids, and we didn't see any of these adverse events leading to a patient being harmed or dying on protocol, which is actually always very, very important when we're looking at these things. Now, the even more exciting thing is that we actually had a 100% overall response rate, meaning that of all the patients we could evaluate, of all 12 patients, every single one of them had a response by IMWG criteria, which is pretty exciting, especially given those patients who had such highly refractory disease. And on top of that, in all patients who were evaluable except for one, they were all minimal residual disease negative, so MRD negative in their bone marrow. And again, that's also really exciting because it means that, within a month of treatment, we basically were to clear the bone marrow of all their plasma cells. And this included a patient who had prior BCMA antibody drug conjugate therapy for a substantial amount of time. And so, when we think about patients who receive BiTEs or ADCs targeting BCMA, a lot of trials don't allow them on. However, we are allowing those patients on this study. What we're finding is that those patients still get benefit from these CAR T cells. So a really exciting thing when we're thinking about how we sequence therapy and what kind of therapies patients should be getting after prior therapies. We also saw that the CAR T cells were persisting at upwards of 4 months in patients, and really, really rapid clearance of disease. So, in summary, I'll just say that this is a novel binding protein for the CAR T cells. It's targeting BCMA. We had about 100% overall response rate, including CRs and VGPRs. We're seeing that these responses are continuing to deepen over time. And we're really excited to be working on the pivotal phase 2 study for CART-ddBCMA, so we can get this into more patients and see how it does in a larger number. So with that, thank you very much, and I hope you found this educational.